Yes.
Okay, so good morning to everyone. Welcome to this CFTC Seminar. For me, a great pleasure to introduce our speaker today, that is Professor Nicolas Vandewalle. For many of you, he's the supervisor of Axel, but in true, Axel is his student from me, right? He's more famous than you. And he was here 2 years ago to give a seminar, and he's back from Liège to tell us the new things from his group. And what I particularly like about these seminars is that he always has some nice experiments that puts us thinking about the physics involved. So thank you very much for being here and looking forward to this.
Thank you for the invitation, Nico. So, as said by Nico, I come from the University of Liège, where I created 25 years ago a group of statistical physics, soft matter, and we started with granular materials, the study of granular materials, then we moved to capillary interactions between particles when we put in the phase, to experiment in microgravity also, and more recently to active matter, and it will be the subject of this talk today. My group today is composed of 12 members, and they are studying the soft matter physics on 4 main axes. First, the capillary interactions, and I gave a seminar 2 years ago here about that subject. Some of them, and Axel is one of the members of this subgroup, is working on magnetic system, and I suppose that he had the opportunity to give some conference to people here about that. Yesterday we had the seminar of Adrien about granular materials, and today I will talk about robotic for active matter. In fact, this subject is very new in my group. We have only a few papers already published, mostly on the technical specification of the robots. And what I will show you today, it's more about what we have done in physics with using those robots, and publication will come by the end of the year, I suppose. The context is, of course, active matter. As you may know, active matter started to grow as a field of physics 30 years ago, roughly. And the subject of this field is to study particles, components, consuming some resources in the environment in order to gain some energy to self-propel, so to induce some motion, or to sustain some complex interactions between these particles, or to exert some forces. And this leads to pattern formation, to swarming collective behaviors, and of course, all those physical phenomena can be found in nature. That's the reason why a lot of physicists try to study living systems, from cells to bird and fish. So the length scale is quite broad, from the micrometer to the meter. And 20 years ago, I was studying fish systems, so I did myself some experiments with tiny fish in big fish tanks. And as a physicist, it is a nightmare. Why? Because the number of particles is not fixed. Because some of the fish may die if you are not feeding them correctly, or they are eating each other. It happens. And so it's really a nightmare in order to collect some data and to get some reproducible data for designing laws or whatever. So Fortunately, physicists are also developing some synthetic materials behaving like living systems, like active fluids, like moving self-assemblies or swimming systems, or by using external input of energy, you can obtain some swarming and collective behavior, which looks like the behavior of colonies of bacteria. And more recently, a lot of groups in the world are studying active matter using tiny robots. And most of the people are using as robots a toy, which is called X-Bug. You can find it on Amazon for a few euros. And they studied the interactions, motion of the X-Bugs, and what we can do in physics with that using the interactions and collective behavior of those things. So, a few years ago, we had the COVID period. So in Belgium, we were stuck at home. And my technicians were at home also, without the machines, big machines, producing pieces for the labs. So I proposed to the technicians to build robots. So that after the period at home, we had a few prototypes of robots. This is based on the stick-slip motion, exactly like for the X-Bug. So you have a tiny motor inside the robot. The robot is vibrating vertically, and because of the legs, which are a little bit inclined, you get some stick-slip motion of this object. Because my technician is very good, I asked him to put some motors in 3D printed parts, but not only a motor, but also a microcontroller, in order to compute the behavior of the tiny robots. Because X-Bugs, you switch on the X-Bug, and the X-Bug is going in one direction, and that's it. I would like to obtain some particular motion, but also interactions between those. And so I asked him to put some sensors, and in the first version of the robots, there was a microphone as a sensor to detect the vibration of the objects. It was also a nightmare. It was a very bad idea because with the sound you have the reflection of the sound everywhere in the lab, and then the robots cannot easily detect others, nearest neighbors. They are detecting the group of robots, and that's it. So we changed a little bit the things, and now we have new versions. And the very last version, which is called the GRASPion, is this guy. You have only 4 legs. In fact, we optimized the shape, the thickness of the legs also, in order to speed up a little bit the motion of the box. You have 2 motors. The motors are here, so the robots can easily go on a line straight if the 2 motors are activated, but it can turn on the left or on the right if you activate only 1 motor. You have a big battery in order to sustain experiment during more than 1 hour. So this is very good. You can charge the system, the battery, using a USB port here. You have a lead here that you can connect on the box. You can change the lead of the box. And on the lead you can place additional power, computing power, of course, processors, or sensors. And by default, we have light sensors. And the sensors can also detect the color of the light from the others. So we have LEDs on those robots. And something strange is that we had some difficulties when we produced the first version of the bots, because, as you remember probably, during the COVID period, in order to obtain some components, electronic components in China, in the US, it was a little bit difficult. And the idea was to use only electronic components produced in Europe. So this robot is purely 100% European robot. So now we have an army of robots like that. We have about 100 robots. So they are charging. We have a dock for charging all the bots. And you can also communicate with the bots using infrared, coming from, for example, from this remote, like for the television. So I will try to activate one bot. I hope it works. And you see the motion, so it will turn here, so you have a carrier motion. And you see a little bit the speed of the bot. Stop it. You can detect also the light. You can compute any kind of motion, so random motion for the turn. So this particle can be diffused, not only ballistic but also diffused. And you can transmit your orders to the swarm of robots using simply this remote system.
Yes, so it vibrates.
Yeah,
it's how it moves. It's the vibration.
It's only the vibration, so there are no wheels. It's just the vibration.
Exactly the speed and the rotational speed also. Or not.
If you increase the rotational speed, you can optimize. You can find some, let's say, resonance for the leg motion, and then you can get probably 10% more speed for the bus. But the most important part is the ingredient for the motion is the inclination of the legs and the flexibility of those legs. So whatever the frequency of the so we studied that kind of physics a lot in order to optimize a little bit the speed of the in fact, we are very happy because we started during the COVID period in 2021 with very simple systems, and now we have a system which is very effective, reproducible. Also, this is very important if you want to perform some experiments, and also the processor. That equip the robots is also a little bit more powerful than before. So you can easily compute the interactions between the bots. And I would like to increase a little bit the computing power. As you see, the vertical axis here is the speed of the bots, and of course the Xbox is very fast. It was optimized as a toy, so it is fast, but it is going only on a straight line. And as you see in the dates, a few years ago, the company was bought by another one, and they are not produced anymore. So you can maybe find on Amazon some parts of Xbox, but it's roughly finished. So, but we can provide other labs with all of us if needed. So So what you can do now is, because we are physicists, I'm not an engineer, so it's quite fun to create a bot, but I would like to make some physics with that. We have plenty of projects with master students and PhD students. For example, we place a flexible membrane around the bot in order to obtain the analog of a moving vesicle or a moving cell. And we create some, if you increase the number of cells or moving cells in the system, we create some tissue, synthetic tissue. We also, here, it's another example of the ability of the bot to communicate using the lights and colors. We compute a programmable system in order to synchronize the different colors between the bots. So they are not moving, they are just communicating with each other. So students are computing, they are learning nonlinear physics to do that. We are computing for the moment non-reciprocal interactions between the particles. So, for example, you have two colors. The blues are attracted by the blue but are repelled by the red, and the red, so it's the inverse, so it's quite fun. And also, I started a project about stigmergy, so the creation of traits in some random environment. And today I would like to present only 3 projects, those in red. So the ratchet, so the motion of a cell in a channel, like in a microfluidic device for real biological cells. Synthetic tissues, this guy, and also stigmergy, the trait formation. And I would like also to say that most of the projects are done also in collaboration with other universities, so people from Germany and Netherlands, or the ILD Institute in Zagreb, the University of Liège, and the university, the Catholic University in Leuven, the Flemish part of Belgium. So let me start with the flexible particle. So you place this robot in a membrane. For example, you get a piece of paper, a flexible paper, and then you glue this paper to create a ring. And then when the bot is moving, for example, the bot is diffusing on the horizontal plane, then it touches the paper membrane, and the paper membrane deforms and also move. And you understand that the size of the membrane is very important. It's the relevant parameter of this experiment, because for a very small membrane, the bot is always touching the membrane. So the correlation between the position, the center of mass of the membrane, the vesicle, and the position of the center of mass of the bots are quite similar. You see the trajectories of the dark line for the bot and the blue line for the cell, and you see very strong correlation in the motion of this system. So we have a diffusive, flexible particle in that case, but if you increase, like in this drawing, the size of the membrane, then sometimes the bot is turning inside the cell without touching the membrane, the border. And in that case, the membrane is fixed and is immobile, while the bot continues to move inside the cell. And in that case, it's this situation, you see a strong decorrelation between the motion of the cell and the motion of the bot. You can also characterize, of course, the physics of those behavior by using what we call in active matter the mean square displacement. So the displacement after some time lapse. and you're in the log plot. If you have different slopes. If you have slopes proportional to the time duration, then you have a diffusive behavior. And if it is the square of this duration, so a little bit steeper slope, you have a ballistic motion. And you see that in all cases we have both behavior. At short time we have a ballistic motion because there is a persistent length due to the motion of the robot, and then the robot is turning and then starts the diffusive motion of the robot. So you can study the scaling, and this is currently done by my colleagues in Zagreb. Yeah.
Where does it saturate? It looks like a cage is inside of the box. So.
If you, in the experiment, in the simulation, of course,
yeah,
we can use an infinite arena, but in reality, in the lab, we have a box which is 1 meter wide. So at some point, you will get a saturation. I would like to build a larger arena, but I need more space in the
like.
It's always a fight with colleagues.
You can use the stadium.
So now, if you look at the literature, biophysicists are manipulating motile cells in microfluidic channels. For example, in this well-known work by the Mahmoud and co-workers, if you place a biological cell in a symmetrical, simple microfluidic channel, then you get the motion of the cells along the direction of the channel, and because it's a random process, then you get diffusive motion, 1D diffusive motion. But if you change the shape of the channel with using triangular-like compartment in order to get some asymmetry between the left and the right direction, as a result, what you get, as you see in this movie, you see that the cell is moving from the left to the right. So we try to do exactly the same with our synthetic the border of the systems. And now, if you look at the experiment, so the first movie on the left is the videos from the experiment, and after tracking the envelope of the cell and the position of the bot, you have this video of the tracking of the experiment, and you see that the cell is moving from the left to the right. So my PhD student, he did the experiment a very large number of times, weeks of experiment, by changing the size of the membrane, because it's a very important parameter, as I said before. And what was discovered is that the probability to go to the right is, for a very small system, for a small membrane, is around 70%. So the probability to go to the right. And if you increase the size of the membrane, you see that this probability increase and reach roughly 90%. And even, I will show you later, situation where we have 100% of probability to go to the right. So that's very strange. And in fact, this situation where the motion of the bot is correlated to the motion of the cell itself, for very small membrane, was also inspiring. And then we tried only with the bot, without membrane. And without membrane, what we get is this value of 70% of probability. So the question was why a single bot, a diffusive single bot, is moving to the right with that probability. And the answer is this. By diffusion, the bot is in one triangular compartment is moving toward the edge of the triangle. Once it reaches the edge of the triangle, it has because of the specific shape of the bot tends to follow the edges. And as a result, it tries to go to the corner of the triangle. So the probability to go to the left, if it is open here and open here, the probability to go in the next compartment is a little bit higher than reaching this wall of the triangle, this edge. And this gives this probability of 70%. As you see, with a large membrane, we increase this probability, but you can also track the position of the bot inside the cell. And if you calculate the probability to go to the right or the left, just for the bot, not for the membrane, you get those probabilities, and you reach again the probability of 70%. So that means that for that cell size, the cell itself is moving to the right, but the bot inside the cell is moving to the left and the right. And you see that the Gauss curve separates around some values of the size of the membranes, and this size is typically the size of the membrane when the membrane is a circular inside the triangular shape. Above, you need to contract, to deform, in fact, the membrane in this compartment. And this is very interesting, because now we try to determine the energy, the bending energy of the membrane, because it seems that the membrane has an effect. And so from the movies, from the tracking of the shape of the cell, it's possible to determine the radius of curvature along the membrane and calculate the bending energy of the membrane. And if you plot this bending energy when the cell is moving inside a single compartment, you see here the triangular shape of the compartment. This is the position inside the compartment. You see that depending on the radius of the membrane, you get different bending energies. That's obvious, but you see that on the curve is not symmetric at all. The slope here is steeper than in that way. That means that the force you need to deform the membrane is much higher to go to the left than to go to the right. And that's, in fact, the physical ingredient explaining the motion of the membrane. Also, you can plot the depth of the bending energy, and you see that we have a minimum, of course, when we are at this critical radius, when the circular cell touch the edges of our material. So that's very interesting, and my colleagues from the University of Zagreb did the simulations, and you see the experiment results and the simulations on the right, and it seems that the agreement is excellent, but using simulations you can also change more easily the parameters than in your experiment. You can extrapolate results from the experiment. And what we found doing that is that if you plot now the probability to go to the right as a function of the force exerted by the robot on the membrane, if the force is very high, the robot is able to push the cell in the next compartment easily, but also in the left direction. And so the probability will be, in that case, around 70%. Otherwise, if you have intermediate value of the force, you get only a motion towards the right and not towards the left. And if the force is close to zero, of course, the cell is stuck inside one compartment. So what we did a very simple experiment to change the force exerted by the robot on the membrane. So we measure, in fact, using a spring, the force implied due to the motion of the robot by changing the mass of the system. So we put a small capsule on the robot, and we add some brains in these capsules. So we change the mass, and we measure different forces. It's a linear behavior of the force with the mass, with the added mass on the robot. And what we found, as a function of this mass, or if you use the other axis, the force, you see the expected behavior of 100% of probability to go to the right. Those are experimental data. And if you increase for a specific size of the membrane, if you increase the force, then you get again 70% of probability. And you have movies here showing that for the same size, you have both behavior. Here is going to the left, and here the robot is unable to push the cell towards the left. So experimentally, we recover what we have predicted in this part of the work. So that's quite nice. So that's it for this example.
Ask you something. So, your argument of the 30% going back and 70% moving forward assumes that the active particle, in your case the robot, has enough time to lose memory from the entrance, right?
Exactly.
Okay. But when you see the video, you see a persistence length along the direction of the ratchet that is much larger than the size of the individual cells. My question is what is the feedback of the motion of the membrane on the motion of the robot? Is it increasing the persistence length?
Yes.
Okay.
Why? Because if you have a deformation, maybe you can see that
in the video, you see that it's moving more than. Yeah.
You see that the robot is, you create a small radius of curvature in the membrane, and then the robot is trapped there and continue to move in that direction.
Okay.
So this increase, so you have an excess of persistence length in that case. So you are right, this effect is very important in this.
So imagine that instead of paper you will use a viscoelastic material without the walls.
Yeah.
So you would expect to have also an increase in persistence length in that case. So you, by adding a viscoelastic material, in principle you could enhance the effective diffusion coefficient of the active motion.
Yeah, so the difficulty is to find the right materials for the membrane in experiment.
Yeah.
But in numerical simulation, you can do that quite easily. The simulations have plenty of different parameters. So we start the second example of what we are doing in the lab. It's about stigmergy. It was first introduced in the 50s, 60s by a French entomologist studying the way insects behave. And a nice example is the case of ants. Creating some trails of traces of pheromones that the other ants followed And then you have a very long, very particular path of ants everywhere. And maybe you know also that if there is a loop, the ants are trapped in this loop forever. I think it's called the vortex of the death or something like that for the ants. But you can find this kind of behavior in different organisms. For example, termites, also wasps, because in fact by changing the environment, you can in fact build very large structures like a nest. So this is very important in biology. But for physicists, you have plenty of questions connected to this effect about the memory effects in a random environment, or those particles behave, the diffusive properties, are there some transitions? And I will show you that this has been addressed in the literature recently. I have three different examples. The first one comes from an experiment of Mathieu Labousse. Mathieu Labousse in Paris was studying the bounce of a droplet on a liquid bath. And at each bounce, the droplet is creating waves on the liquid bath. So the waves are changing the environment where the droplet bounces. And this induces some motion of the droplet. So the droplet is not bouncing at the same position. The droplet is bouncing following some specific trajectory. But you can, in the experiment, you can damp differently the waves. So the persistence of the waves along the liquid interface. So you can play with the memory effect in the system. And if you change the memory, this parameter, you change the path from a deterministic simple path with trefoil trajectory towards chaotic trajectory. So that's very interesting. Just by changing the environment and the way you interact with your environment, you change, in fact, the trajectory. A second example comes from the group of Muñoz. They studied active colloids in passive ones, a huge system composed of passive ones. And as you see here on those pictures, you see that the active particles are following the path created by the others, and at the end of the story, they gather and they are forming some clusters, tiny clusters in the systems. Meaning that there are some memories created in those systems. And more recently, people did the similar experiments, but using the exosomes. So tiny robots in environments where the robots are pushing small particles. Small particles are shown in this picture in white. And you see the trail of the robots in those systems. There are many questions on those works. And of course, there are many parameters and many experimental conditions to study the effect of trail formation. But what we did in my book was to study the effect of the environment on the trail formation, and we decided to change the packing fraction of the particles. So we have tiny blocks, so 1 centimeter by 1 centimeter blocks of wood, and we place a quantity of blocks covering 30% of the arena here, 50% and 70% of the arena. And you see that whatever the packing fraction in the system, you get trail formation. You see that there are some voids behind the motion of the bots. You see the bots everywhere? Yeah? Yes. And that's very interesting because you see that there are two main observations. The first one is that the speed of the bot is the same. So the program is exactly the same in the microprocessor of the bot. But due to the higher packing fraction, the bot has some difficulties to push the tiny blocks. So the speed decreases a lot between this situation and this situation. The second observation is that here, if you look specifically at the trail, I can speed up a little bit. You see that you erase, the bot is erasing the previous trails. But in that system, you see that the bot, once some trails are created, the bot tends to follow always the same kind of path due to the high packing fraction. And if we look at very long times, more than 1 hour of experiment, you see that the green loop, so the bot is following its own path, and it's exactly like the vortex of the death for ants. So it's a similar effect. So we studied, in fact, how the trail emerged in the system. So it's quite easy. You cut your arena in squares, and then in each square you measure the packing fraction. So you have a measure of the local packing fraction in the system. Of course, my PhD student is placing randomly the blocks at the beginning of the experiment by hand. So even if the packing fraction is 5%, at the beginning of the experiment, the local packing fraction is a broad distribution. Okay? But if you look now, the situation at the end of the experiment, after 1 hour of motion of the bot, you recover exactly the same kind of distribution. So it's the same broad distribution. So nothing particular happened in that. But if you increase the packing fraction, you still have a broad packing fraction, which is PhD equivalent. If I change my PhD student, probably I will get another student. But so it's the same person doing the experiment here. And if you wait 1 hour, then what you see is the formation of the trails here, so the voids or channels in the system. And you see also a shift of the second peak, this peak. So your second peak appears at high packing fraction, meaning that you push the blocks and you form aggregates. And in fact, the bot is never going to that part of the system. So that's interesting.
Sorry, can I just ask something? If in the beginning you are putting the pieces at random, then why is it that at the edge it seems that we never have...
Yes, good question.
Is
in
it
fact,
because
we measure only the local packing fraction in the central part of the arena. Because the bots are always going to the edge of the arena, and we are using this particular shape, a flower shape, in order to resend the bot inside the arena. So those effects are neglected in this study, so we measure only the local packing fraction in this part of the arena.
But in the beginning you put the wooden pieces like close to the edge?
No,
never?
Just in this part.
Ah, okay.
Yeah.
Okay.
In order to avoid the global change of the packing fraction, of course, during the experiment. What the PhD student did also